సార్
దాంట్లో గ్రూప్ మ్యాథ్స్ దాంట్లో పెట్టారు కదా వాట్సాప్ లింక్ అలా పెట్టండి కానీ వాట్సాప్ గ్రూప్ క్రియేట్ చేస్తారు 1 2 అని పెట్టారు కదా అలా పెట్టేసి గొడవ చూసినప్పుడే
కటింగ్ हाँ आएंगे हाँ खा अब सीधा खा बड़े हैंडी कर हाँ कल टाइम को 11:30 होता तो मैं 5:30 बस निकला ये 1 आवर बिफोर आना हो 9:30 का तो नाश्ता ऐसा करो तो कर डालिए कब अच्छा लगे 6 बजे से 6 बजे होता क्या समझो ना मेरी इंदिरा इंदिरा एसी दूसरा अब रखो आपने मेरे दिन अभी हो नहीं के बारे में कॉल करी मां कब पे मैं राजस्थान में आती तक ना होता आओ फिर तो उसमें क्या है घर में सही फुल है सबका है अरे घर ऐसे सो घरा से आया थी बोला तो मैं बोल तो मेरे को भी आ सकती क्या है वीडियो
वसमान भाई वॉइस बंद करो
इंशाल्लाह
समय से पहले ऐसा नहीं कर सकते आप कहीं और तो निकल भी हो
Hello. Madam, good morning.
ఈయనంత చేయకూడదు. Good morning, all of you. Okay, na? President of our college management, Sri Petan Surnarayan, Secretary and Correspondent of College Management, Dr. Ramchandra Raj garu, Treasurer Sri Kumar garu, and the speaker of today's webinar, Assistant Sir, Guru, Head of English, Dr.K.S.
Recording.
And convener of this webinar, Dr.K.S. Faculty who have participated in this webinar from hello, Praveen garu.
Hello, sir.
Good morning. Seeing you after long
Sir,
time.
voice. Sir,
Voice slow ga unda.
yeah, little bit కొంచెం స్లోగా ఉంది సార్, నెట్వర్క్ మీ సైడ్ డౌన్ ఉన్నట్టుంది.
Voice. Okay. Praveen garu ala unte
అటు సైడ్ డౌన్ ఉంటుంది.
Not audible, sir.
Hello, sir.
Not clearly audible. Hello, good morning, sir. Not clearly audible, sir.
Network is not good, sir. Wait a minute.
Hello, sir. Is my voice is audible?
Yes,
Okay,
sir.
nice.
Yes, sir.
Audible.
Right.
Yes, sir.
Sir, do you want me to start, or is there any introductory program, something like that? Because I'm not able to hear your voice.
Wait a minute, sir. Wait a minute.
Okay, okay, right. ఇది క్లారిటీ ఉంటుంది, క్లారిటీ ఏ క్లారిటీ
అది కూడా తీసే కొంచెం అందరూ ఎదురు పెట్టారు కొంచెం.
Sir, your voice is not audible, sir.
Hello.
Hello, sir.
Sir, you are talking about me, sir. My voice.
No video, sir. Only audio is available, sir.
Good afternoon, sir.
Number can't be reached right now. ఈ నెంబర్ని ప్రస్తుతం
సార్ స్టార్ట్ చేయొచ్చా, వెయిట్ చేయాలా?
Sorry for the. Thank you for sitting. In the field of physics, mathematics, electronics, and computer science, we hear the term, we learn the term, we study the term vectors, where any quantity, it may be a physical quantity, which has magnitude, that means value, and direction. It's called a vector. For example, considering the physical quantities, displacement, velocity, acceleration, and we know the force. These are all vector quantities. Considering alternating current, we have been using. Without it, we cannot conduct this webinar. With contrast to direct current, so alternating current has a value and direction. So alternating current AC can be considered as a vector. So coming to biology, vector is a Latin word which means to carry. That means vectors are carriers that transmits diseases between individuals. Mosquitoes and flies are the examples of vectors. So these insects transmits parasites between the individuals, and the causes we know, dengue, the malaria, like that. And animals which carry infecting agents from an infecting individual to an healthy individual. So insects cause diseases which transmits parasites from one body to another body, same as with animals. And if we consider humans, so humans also can say the examples of vectors. We during the period, human transmitted this COVID virus from his body to another individual. So the topic of technology. Of DNA, which case transfers a gene or a genetic material, usually gene, from one cell to another cell. So there must be a purpose regarding this transportation of genes from one cell to another cell. So the purpose may be multiplying the genes or isolation of genes. So their simplicity may be in the structure and rapidity of the process. These vectors are very much used in molecular biology and genetic engineering. And among all the applications, the other important application of one of the applications of vectors is we can study the structure, function, and production of a protein in different organisms. So with these few words, I am concluding my remarks. Thank you very much for your patience listening, the participants. Moving to the next, I invite IQAC coordinator and head of the Department of English, S.V.K.P. garu.
అది ఒకసారి చూస్తే మనకి ఈజీ అయిపోతుంది. అంటే వీళ్ళు ప్రిపేర్ చేసి పెట్టినారు అని చెప్పేసి ఇక్కడ కామెంట్ చేస్తున్నారు.
Audible? My voice is audible.
Sir, excellent, sir.
Yes, good.
For which?
Good morning, good morning, participants. Respected chairman of the webinar, our dynamic principal, Dr. Y.V. Apparao garu. We have to express sincere greetings to our college management, President S.V.K.P. garu, Secretary and Correspondent Dr. K. Ramchandra Raj garu, Treasurer Sri Uddagiri Lavkumar garu, for their valuable suggestions, their guidance in all activities of the college. First, I convey my comments and greetings to the convener of the webinar, Dr. K. Lakshmikantam garu. I'm really appreciate with the support of the biotechnology department. She's also designed the program within short period. I'm really congratulate faculty members of the zoology and the biotechnology. My sincere thanks to all the participants who are participating the webinar from various places. It is also very helpful to you. Finally, I convey my sincere thanks to Sri Pravin Dutt sir, who is my brother, formerly who's also worked in our college for a period of time. I'm really thankful to you. At the same time, hearty welcome to Pravin Dutt sir Once again, we would like to enjoy the sweet lesson of yourself. My hearty welcome you. At the same time, all the participants, we have to equip the good knowledge, sharing the company of Pravin Dutt sir. Hearty welcome. Thank you all for giving the wonderful opportunity. Handed over to our principal sir.
Thank you, Shash Kumar, for your wishes on the occasion. Now I invite convener of this webinar and head of the Department of Zoology UG courses to introduce our guest speaker, Dr. Pravin Dutt garu, to the participants.
On behalf of S.V.K.P and Dr.K.S. Arts and Science College Management, Principal, staff and students, I extend a warm welcome to you all to the national webinar on vectors and their role in biotechnology, jointly organized by Departments of Zoology and Biotechnology in association with IQAC. In this occasion, I would like to introduce today's resource person of the webinar, respected Dr. Pravin Dutt garu, to the participants. Dr. Pravin Dutt is a gold medalist in MSc Zoology from Andhra University, Visakhapatnam in 2004. He was awarded PhD from Andhra University in 2019. It's a great privilege for all of us that he worked in our S.V.K.P and Dr.K.S. Arts and Science College in the Department of PG Zoology for over a period of 2 years. He got state 5th rank in JL selections in 2008. Later, by virtue of services, he was promoted as a degree college lecturer in 2013. To his credit, he had 3 publications. Ever since, he is a Sakshi columnist and wrote 50+ articles in Sakshi Daily on various competitive examinations, especially for Group 1, Group 2, civil services, and on various issues like environment, biotechnology, genetics, science and technology. He delivered lectures in Mana TV programs. He is a content generator and subject expert for many LMS videos. He is a podcaster and produced podcasts on biology. He rendered his valuable services in various corners of the state like Kamampadu, Nuzividu, Chintalpudi, Mailavaram, etc. At present, he is working as assistant professor in the Department of Zoology in Government Degree College, Tiruvur, NTR district. With these few good words, I humbly present Dr. Pravin Dutt garu to the participants. Now I request our beloved principal and chairman of today's webinar, Dr. Y.V. Apparao garu, to continue the proceedings. Thank you, one and all.
Thank you, Madam. Dr. Lakshmi Kantam garu, head of the Department of Zoology of our college and convener of this webinar. Introduction of guest Dr. Dutt garu. Thank you, Madam. Now I invite Dr. Pravin Dutt garu to the webinar and continue.
నేను చాంబర్లో ఉంటాను. మీటింగ్ ఎన్ని గంటలకు అన్నారు? 3గా. నేను చాంబర్లో ఉంటాను. ట్రెజరీ పోయే ముందు వచ్చాను.
Right, sir. Thank you very much for your welcoming words. Dr. Raju garu, correspondent of this college, and Dr.
Pravin garu.
Sir, is it audible right now?
Yes, sir.
Right. Good morning to you all.
Morning.
And respected Raju garu, correspondent of this college, and Dr. Apparao garu, principal of this college, and Dr. Shashikumar garu, the IQAC coordinator, and Dr. Lakshmi Kantam garu, head of the Department of Zoology, and Dr. Ramesh garu, head of the Department of Biotechnology, and my dear colleagues, Dr. Pullarao garu and Satish, and all my dear colleagues of S.V.K.P College. Thank you for this opportunity, and I'm very glad to see all my former colleagues. I'm so happy to be a part of this seminar. And the students participating in the group, I want to be more responsive. Now and then I'll ask questions and I'll make it this more interactive. So I'll be asking some questions, or sometimes I ask you to type. So you be respond to the chat box, right? Can I present the PPT, ma'am? Kindly allow me to present my PPT. Please permit me share the screen. Please permit me share my screen. Hello, sir. Right. So vectors, as our principal said, we use the word vectors in different subjects. In physics also we use the word vector. In zoology we use the word vector, but the meaning is different. And in biotechnology also we use the word vector. So in this webinar, I'm going to focus on the cloning vectors. What are their characters of vectors and different types of vectors, and how they are useful to mankind. Before I go into the detail, I would like to grant your attention to know about what is genetic engineering. Genetic engineering is a process of joining two different DNAs and make a hybrid DNA is called genetic engineering. In this in this picture, you will see a glowing glowfish. This is called a glowfish, where they produce light. So this glowfish is a genetically modified fish. We take the DNA of jellyfish, which produce light during night time, and we take the gene from the jellyfish and we placed into the fish. So now this is called a genetical hybrid, and the technology we use is rDNA technology, recombinant DNA technology. We use two different kinds of DNAs, we merge it. Hence, this is called recombinant DNA technology. So in this, the first step is isolation of target gene. See, in order to produce this glowfish, first of all, we have to isolate the gene from the jellyfish, because jellyfishes, they produce light during night time, different colors of light. We pick up the gene that is responsible for light, that is called target gene. We isolate it. That is step one. And the step two is we transfer that particular gene into the fish or host cell. There we use the word vector. How to transfer genes from one organism to another organism. So for the purpose of transferring genes from one cell to another cell, we use vectors. We need vectors. Recently, entire world is affected with COVID. And the scientists prepared COVID vaccine. How do they develop the COVID vaccine? Here, with the help of vectors, they introduced that COVID virus protein coat into a bacteria to produce vaccine. The scientists pick up the COVID genes and they transfer that COVID gene into a plasmid or into a bacteria For that purpose, we use vectors. Means vectors are the vehicles that carry genes. So before I go into the definition, I would like to give you some example. I think all of you seeing this picture, right? Here a father holding a son on his shoulders. Now I can simply say that this person is a vector, and the boy on the top is a target gene. What do you understand by seeing this picture? The vector carry target genes. Simple. Vectors, they carry the target genes. I'll show another picture. See here, you see a vehicle, right? You see a vehicle here, Jeep, and it is carrying few people there. Like how? Like this. Vehicles carry people, whereas vectors carry genes. Do you understand what I'm saying, my dear students? If you like this example, please type wow, W-O-W, wow. All of you type wow. Do you understand? The picture itself indicates the vectors means they're the vehicles that carry genes. Vectors are the vehicles that transfer genes. That is the simplest definition that I can give it to you. Now let's go to the definition of a vector. Vectors are the vehicles which are used to transfer desired genes into a host cell. And vector itself is a molecule of DNA. And these vectors can have a capacity to replicate on their own self. In other words, vectors, they carry DNA, hence they are called DNA carriers. So let's look at the characters of vectors. So first of all, the vector must able to carry DNA. It must able to transfer the DNA. Number two, it must have ability to replicate in the host cells, self-replicative. And the third one is a vector must have multiple cloning sites. A vector must have multiple cloning sites, and it should contain marker genes, selectable marker genes, specific marker genes, we call it is, like tetracycline. And vector should have an origin site, and it should have a high copy number. I am limiting my PPT especially to the needs of the students. And the another character of vector is small size, less than 200 kb, with a low molecular weight, and it is must be easy to isolate and purify. And the vector must contain promoter and operator. So these are the few characters of vectors that we use in biotechnology. So what are the uses of cloning vectors? There are many uses. The first one is to clone the DNA, to produce multiple copies of a gene. We use these vectors. To produce genomic libraries, we need vectors. To prepare DNA probes, vectors are needed. And in all genetic engineering experiments, we need vectors. And very important is vaccine production. For every vaccine, we use vectors. Without vectors, you cannot produce a single vaccine. Even the latest recent COVID vaccine also produced with the help of these vectors. And there are many more uses. What are the uses through the DNA, RDNA technology? Production of human enzymes like human insulin. For that production of human insulin also we are using vectors. Production of all kinds of human hormones and human enzymes, we need vectors. To produce genetically modified organisms, genetically modified plants, genetically modified animals, to produce all these things. And in all genetic engineering experiments, we need vectors. Now, let's see the classification of vectors. On the basis of gene of interest, vectors are of two types, the cloning vectors and expression vectors. On the basis of host cell, there are vectors for bacteria, vectors for yeast, vectors for animals, vectors for plants. And on the basis of cellular nature of vectors of host cell, vectors of two types. One is prokaryotic vectors and eukaryotic vectors. If you look at the prokaryotic vectors, we see one by one. Plasmids, bacteriophages, cosmids, and viral vectors. These are all the prokaryotic vectors. And we have also eukaryotic vectors, where we use yeast cells, yeast vectors. And we use separate vectors for animal cells. For example, in animal cells, we use baculovirus as a vector for animal cells. Bovine papilloma virus, it is a vector for animal vectors. S.V.K.P, that is a virus-based vectors for animal cells. We cannot use the same vector for prokaryotic cells and eukaryotic cells. That's the reason why we use different kind of vectors for animal cells and plant cells. So there are some vectors for plants, they're called plant vectors. And we also use artificial chromosomes like BAC and YAC, bacterial artificial chromosome and yeast artificial chromosome. So these are the vectors used in eukaryotic cells or eukaryotic organisms, because there is a lot of difference between prokaryotic cells and eukaryotic cells. The promoters and operator regions of eukaryotic vector are not feasible to work in eukaryotic cells, because they have a different promoter region, different operator region, which does not match with eukaryotic cells. Right. Now let's look at the prokaryotic vectors. As I told you, prokaryotic vectors are very important, especially for the syllabus for the 4th semester students. These are the major part of the syllabus. Prokaryotic vectors. First of all, we'll study the plasmids. So what are plasmids? Where they are present? Basically, plasmids are present in bacteria. Here in the picture, you will see a bacteria. And this brown color thread-like structure is the DNA of the bacteria. Besides this DNA, there is an additional DNA in blue color, extra DNA, another DNA. And this extra DNA, which is round in shape, they are called plasmids. So plasmids are, by looking at the shape, you understand they are circular. By looking at this picture, you understand they're double-stranded and they're extra chromosomal. The word itself indicates the extra chromosome, means this is the real chromosome. In the picture, bacterial DNA is a real chromosome of the bacteria. This is. In addition to this, there is another genetic material. It's called extra chromosome. And they are self-replicating DNA molecules present in bacteria. They are called plasmids. Remember, vectors are also DNA molecules, and they carry another DNA molecule. So here, plasmids are extra chromosomal molecules. In this picture, you can see these plasmids are present in cytoplasm of the bacteria. So this round color one is called red color, and round shaped one is called plasmid. And this is the original genome of bacteria. In addition to this DNA, another DNA is present, is called plasmid. And the number of the plasmid may vary from cell to cell, from sometimes single plasmid to the hundreds of plasmids. The size of the plasmid also vary from 1 kb to 500 kb. Right. Types of plasmid. There are two types of plasmids. Now, number one is natural plasmid. The second one is artificial plasmid. Natural plasmids are naturally available in bacteria. Whereas artificial plasmids are, they're man-made. They're man-made plasmids are called artificial plasmids. Natural plasmids are naturally collected from bacteria. They are large in size, and they have the genetical markers are absent in natural plasmid. This is very important. And there are no common restriction sites in natural plasmids. That's the reason why natural plasmids are not suitable for genetic engineering. Natural plasmids are not suitable for genetic engineering. That's the reason why we produce artificial plasmids. We just modify the plasmid. We take the natural plasmid from the bacteria, we delete some genes, and we add some genes. That's how an artificial plasmid is made to suitable the needs of genetic engineering. So artificial plasmids are generally small in size when compared to the natural plasmids. And artificial plasmids contains separate sites called genetic markers. They are present in artificial plasmid. And the common restriction sites are present in artificial plasmids. That is the reason why artificial plasmids are most suitable for genetic engineering. For all the genetic experiments, we use only artificial plasmids because natural plasmids are not at all suitable for genetic engineering experiment. You take anything like production of vaccine, production of enzymes or hormones, to produce DNA libraries, whatever it may be the purpose, to produce genetically modified organisms, whatever may be the purpose, we use only artificial plasmids, which are made from natural plasmids by deleting some genes and adding some genes. Right. So plasmids are again of two types, that is conjugative plasmids. Plasmids again of two types, that is conjugative plasmids and non-conjugative plasmids. In conjugative plasmids, conjugation takes place between the bacteria. So here in this picture, you see the bacteria which have plasmid are called a donor. The another bacteria, they do not have plasmid, they are called recipient. So now a pilus is formed between these two. Conjugation is taking place. The bacteria from the donor, the plasmid from the donor bacteria is transferred into the recipient bacteria. So this is called conjugative plasmids. So in conjugative plasmids, it facilitates conjugation, and they contain progenes which are present in conjugative plasmids, whereas absent in non-conjugative plasmids. Conjugative plasmids are large in size. Non-conjugative plasmids are small in size. Conjugative plasmids, they have got a tough control of DNA replication, stringent control. In case of non-conjugative plasmids, relaxed control of DNA replication. The conjugative plasmids are low in number, whereas non-conjugative plasmids are high in number. And we can also, the another classification is fertility plasmids, resistance plasmids, call plasmids. These are another type of plasmids. Fertility plasmids, they perform conjugation. Resistance plasmids, they carry genes for the resistance for a particular antibiotic. And call plasmids, they carry genes that produce protein and kill bacteria. Now let's look at the nomenclature of plasmids. Like every person has a name, like every place has a name, every plasmid has a name. But there are some rules must be followed to give name to a plasmid. The name of the plasmid must begin with letter small p, p for plasmid. And it should be followed by the first letter of the person who created it, who identified it. For example, if I construct a plasmid, I can put,
sir.
Yeah.
Please click on the record button, sir, once.
Record. Record to the cloud or record to the computer? Record to the cloud or computer?
Thank you, sir. Thank you, sir. Continue, sir.
Right. Okay. So if you name a plasmid, so the plasmid's first letter will be taken from the person who created it. For example, my name is Praveen. If I invent a plasmid, my first letter in my name is P. It must be there in the plasmid. If a person named Kiran constructs a plasmid, the first letter in that name of plasmid must be K. And it must be followed by number given to that strain. For example, let's look at PBr322. This is one kind of plasmid, PBr322. This is the most popular plasmid all over the world. And it's the first of kind of its artificial nature of plasmid. That's why PBr322 is the name of a plasmid. It's called mother of all plasmids. And it is the first artificial plasmid. That's the reason it's very popular, especially used for E. coli, Escherichia coli. In that P, small p for plasmid. B and R, this plasmid is constructed by two scientists named as Bolivar and Rodrigue. The first letter in the Bolivar, B, Rodrigue, the first letter is R. So these two persons constructed this plasmid. That's the reason why the first letters are taken from their names. And 322 is the number given in the laboratory, clone number. And this plasmid contains 4,361 base pairs. And it is a circular double-stranded DNA. And this plasmid contains 528 restriction sites. And there are 66 restriction enzymes that can cut this plasmid. And this plasmid contains restriction sites 528 for 66 restriction enzymes. As I told you, plasmid must contain genetical markers. There are two genetical markers, antibiotic resistance gene markers, we call them as, for tetracycline and ampicillin. TET4 gene that provides resistance to the antibiotic tetracycline, whereas AMP gene provide resistance to antibiotic ampicillin. And it must have an origin site called ORI. Many restriction site enzymes like E. coli, Kind3, BAM, these are the different restriction enzymes that can cut this PBr322. In this picture, you can see this is the picture of PBr322. So here this blue color is tetracycline resistance gene, and the green color is ampicillin resistance gene. And a plasmid must have an origin site, origin of replication. And it must contain restriction sites for different restriction enzymes, like EcoRI1, Kind3, BAM-H1. So these are all different restriction enzymes, restrictive sites. So what are the advantages and disadvantages of PBr322? PBr322 is a small in size and easy to purify. Two selectable markers are present. Can be amplified up to 3,000 copies. These are the advantages of PBr322 plasmid. It has disadvantages too. It has a very high mobility, and it cannot transfer DNA fragments more than the length of 15 kb. 15 kb means kilobytes. And the second plasmid is PUC plasmid. As I told you, P for plasmid, U for UNC means University of California, because this is invented by the researchers of California University, University of California. So first letter of University U and California C taken and named it as PUC plasmid, because it is constructed by scientist and his team, Messings and his colleagues in 1983. Because of their love for their university, They named it after their university, University of California. So there are many series of plasmids like PUC 8, PUC 9, PUC 12, PUC 13, PUC 18, PUC 19. There are different types of PUC plasmids. Generally, what you see in the picture is PUC 19. And this PUC plasmid is prepared from PBr 322. PBr 322 is the first one to prepare, and it was modified and produced PUC plasmid. And its size is 2,676 base pairs. The specialty of PUC plasmid is has an additional gene called LAC-Z gene, LAC operon. You heard about LAC-Z gene. This is in addition to the PBr 322. We can see this presence of this gene. And the remaining is ampicillin resistance genes are present, origin site are present, and the common restriction sites like EcoRI, HIND3, BAM3. These are all very common restrictive sites for PUC plasmid. And the second one is bacteriophages. Here you see in prokaryotic vectors, the first one is plasmids. The second one is bacteriophages. Right. So what is a bacteriophage? Bacteriophage is a virus that infects bacteria. It's discovered by Frederick Twart and Felix in 1917 from France. So these viruses generally they eat bacteria, they destroy bacteria. That is the reason why the word bacteriophage means phage means eating. Bacteria means bacteria. Means this virus they attacks bacteria. They eats away the bacteria. Bacterial eater, it is called bacterial eater. That is the reason why it is called bacteriophage. Phage means to eat, right? That's how the name is coined. It is also called a lambda phage. It's a virus of E. coli virus. In this picture, you can see this is a structure of a bacteriophage. It has a head region. This head region contains DNA, and this one is polar, long tail, and some binding proteins and base plate. The genome of bacteriophage, it is a linear genome. Remember, in the plasmid and bacteria, the genome is circular, round shape, right? Double stranded. Here, the DNA is linear, straight, means it is not circular. Of course, it is double stranded. And the size of the DNA is 48.5 kb with the 12 nucleotides. This is about bacteriophage. And this contains COS sites. And these vectors, the purpose of vector is to carry genes. Plasmids also carry DNA up to a length of 15 kb. Bacteriophage also carries genes with a length of up to 24 kb. Means they're better than plasmids. They can carry lengthier DNA than plasmids. So we use bacteriophage as a vector. You know, if you look at the genome of bacteriophage, if you look at the DNA of bacteriophage, there are two regions. In the picture you see here, this is the DNA of bacteriophage. There are two regions, essential region and a non-essential region. Two regions are present in the DNA of bacteriophage. The name itself indicates essential, atyavasaramaina, avasaramaina, upayogamaina. Non-essential, avasaram leni. So the essential region of the bacteriophage DNA contains genes for head region, tail region, and factors of recombination and lysogeny. Very important activities of this bacteriophage are regulated by essential region. They contain genes for head of the bacteria, tail of the bacteria, and replication of the bacteria, recombination of the bacteria. Whereas a non-essential region, it is located in the middle of the essential region. Here on both sides, essential region is present. And in the middle, non-essential region is present. And now you can replace this non-essential because it is no more use. ఇక్కడ కనపడుతున్న బ్యాక్టీరియా ఫేజ్ డిఎన్ఏలో ఉపయోగపడేటువంటి డిఎన్ఏ ఉంది, దాని ఎసెన్షియల్ రీజన్ ఉంది. ఉపయోగపడని డిఎన్ఏ కూడా ఉంది, దట్ ఈస్ కాల్డ్ నాన్ ఎసెన్షియల్ రీజన్. Now we use this non-essential region to transfer genes. You can place some other gene in this non-essential region, or you can separate this non-essential region and you can keep the target gene. మనం ఏ జీన్ అయితే ట్రాన్స్ఫర్ చేయాలనుకుంటున్నామో ఆ జీన్ ని నాన్ ఎసెన్షియల్ రీజన్ లో పెట్టవచ్చు. అలా పెట్టడం ద్వారా ఆ నాన్ ఎసెన్షియల్ రీజన్ లో తీసివేసి దానికి బదులు దీన్ని మనము తీసుకెళ్లవలసిన జీన్ ని దీనిలో ప్యాక్ చేయడం ద్వారా దీన్ని జెనెటికల్ ట్రాన్స్ఫర్ ఎక్స్పెరిమెంట్స్ వాడతాం. అందుకని లాంబ్డా ఫేజ్ ని మరలా రెండు రకాలుగా విభజించవచ్చు. దర్ ఆర్ టూ టైప్స్, దట్ ఈస్ ఇన్సర్షన్ వెక్టర్స్ అండ్ రీప్లేస్మెంట్ వెక్టర్స్. Insertion, the name itself, the word itself indicates to insert. Replacement means replace. Here in the picture, first of all, let's see insertion vectors, where the desired gene can be inserted into the middle of non-essential region. Here in the picture, you can see this is a non-essential region, no use. Here the target gene. If you want to insert any gene or target gene, now you are inserting it in the middle of the non-essential region. You cut the non-essential region in the middle with the help of restriction enzymes and you insert the target gene. ఏ జీన్ అయితే ట్రాన్స్ఫర్ చేయాలనుకున్నామో దాన్ని ఈ నాన్ ఎసెన్షియల్ ప్రాంతంలో ఇంట్రడ్యూస్ చేస్తాం, చొప్పిస్తాం, దాంట్లో ప్రవేశపడతాం. We insert it, and later this virus is allowed to infect bacteria. Like how the genes are inserted by using bacteriophage DNA. What you see here is the DNA of bacteriophage. They are called insertion vectors. In this process, you can transfer DNA a length of 35 kb. 35 kb length of DNA can be transferred by insertion vectors. Plasmids carry 15 kb DNA. Bacteriophage carry 24 kb up to. Now these insertion vectors, they can carry 35 kb, much bigger and lengthier DNA. Replacement vectors. The word itself, replace, we remove it and we place it with somebody. The non-essential region is removed completely and it replaced with a foreign gene or the desired gene. Here in the picture you can see, this is the DNA of lambda phase. The red color is essential DNA. The middle one is non-essential DNA. That is a replaceable region. We remove it completely and we introduce the target gene, new one. That is why here I showed with the two different colors. We remove the non-essential region and we replace it with the target DNA or the target gene, so that now this DNA can carry the DNA. So these kind of vectors are called replacement vectors, or they are also called substituent vectors. They are also called substituent vectors, and mostly they allow to infect E. coli bacteria, and we allow them to take their life cycle like lysogenic, so that genes are transferred to bacteria. And another vector is M13 phase vector. Here is M13 phase, that's a filamentous bacteria phase used. It is also a virus that affects E. coli. It has a protein coat called a capsid, where the DNA is present inside the capsid. And the DNA of virus is circular, single stranded. Remember, the DNA of bacteria phase is linear and double stranded. Whereas M13 phase, the DNA is circular and single strand. Let me show the difference clearly. Here I talked about bacteriophage DNA is linear, straight, double stranded, right? If you see the M13 phase vector, the DNA is not linear, it is circular DNA. It's not double stranded, it is single stranded. You make a difference between the lambda DNA, that is bacteriophage, and M13 phase. Both are viruses, both are vectors, both can carry genes, but the difference is lambda phase DNA is linear and double stranded, whereas M13 phase virus DNA is circular and single stranded. Here in the picture, you can see M13 phase vector. Here, the single stranded DNA is present, covered and protected by protein coat. So after M13 phase enter into E. coli, the single stranded DNA becomes double stranded DNA and useful for replication. Now it is called replicative form, RF form. It is called RF form, replicative form, which contains short intelligence sequence, IS sequence, which is used for gene cloning. Got it? So M13 phase vector, it contains single stranded DNA. But when this virus infects bacteria, the single stranded DNA becomes double stranded DNA. And now it is called replicative form, which contains special sequence called short intergenic sequence. And this short intergenic sequence is used for gene cloning. Now this region can be used to transfer genes. IS sequence, it is called IS sequence, constructed by Messings and colleagues. And the third one is cosmids. Cosmids means, so cosmid is a combination of plasmids and bacteriophage. As I told you in the bacteriophage, there are special sites called COS sites. COS sites are present in bacteriophage. And we take that COS sites from bacteriophage and we merge it with plasmid. COS sites, they are present in bacteriophage. I can show here. Yeah, in the bacteriophage, here the one point is, on both ends of the genome, a pair of special sites are present, which are called COS sites. So COS sites are present in bacteriophage. We take that COS sites and we merge it with here. This is the COS sites of bacteriophage. We merge it with the plasmid, and it becomes cosmid. So we have different vectors. You got plasmids, they can carry 15 kb. Bacteriophage as a vector, 24 kb. M13 phase, 35 kb. And now cosmids, 45 kb. You can see increase in the size of the gene. For example, if you want to move from Palakollu to Narsapuram, it's a matter of 25 kilometers, right? You can go by bike if 2 people are there. If five or 4 people are there, they can go by an auto or a car. If 20 people are there, they can go by bus. If 100 people are there, they can go by train. See, we use different vehicles with increasing in the number of people. More the number of people, bigger is the vehicle. Small the number of people, smaller is the vehicle. Similarly, if you want to transfer small fragments of DNA, small genes, we just use plasmids, like 15 kb. If you want to transfer much bigger DNA, we use bacteriophage. Still bigger DNA, we use M13 phase. Still bigger DNA, we use cosmids. So we use different kinds of vectors based on the size of the gene that we are going to transfer. మనం ఏ జీన్ అయితే ట్రాన్స్ఫర్ చేయాలనుకుంటున్నామో ఆ జీన్ యొక్క సైజును బట్టి మనము వెక్టర్స్ ని సెలెక్ట్ చేసుకుంటాం. Here the cosmids, they can carry up to 45 kb. The length of the DNA is 45 kb. They can carry that much lengthier DNA up to a size of 45 kb. So cos sites are special sites present in the DNA of bacteriophage. They are sticky in nature, cohesive ends. So that is about cosmid. If you look at the cosmid, here, as I told you, cosmid is a combination of a plasmid and a bacteriophage. Here, these three characters are the plasmid characters of plasmid present in the cosmid. ఈ cosmid లో ఉండే plasmid లక్షణాలు, ఈ cosmid అనేది plasmid మరియు bacteriophage రెండు కలయిక వల్ల ఏర్పడుతుంది. కాబట్టి ఈ cosmid లో ఉండే plasmid లక్షణాలు ఏంటంటే ఆరిజిన్ సైట్ ఉంటుంది, యాంటీబయాటిక్ రెసిస్టెన్స్ మార్కర్స్ ఉంటాయి, multiple cloning sites, and the cos sites are taken from bacteria. So with this phenomenal combination, the cosmids are made. So these are all prokaryotic vectors. We have also some animal vectors where we use baculovirus, baculovectors, baculovirus vectors. As I told you, there are separate vectors for both plants, animals, separate vectors for prokaryotes and eukaryotes. In the beginning, I showed a here these are the eukaryotic vectors, whereas we use yeast bacteria in eukaryotics. Vectors for animals, we use baculovirus vector, bovine papilloma virus, S.V.K.P virus. These are the viruses which are used as vectors to infect animal cells. Similarly, we have plant vectors too. Yeah. So there are many animal vectors, but I am focusing only on baculovirus and adenovirus, which is most popular, and especially adenovirus is used to prepare a COVID vaccine. So here, baculovirus, it infects insects only. This virus is rod-shaped with a large double-stranded genome. The gene of interest is expressed during the infection and the very high yields of protein. So this is the baculovirus generally we use as a vector for animal cells. Now, I would like to draw your attention for adenovirus. Adenovirus as a vector, especially for the mammalian hosts. Adenovirus is widely used, to prepare COVID vaccine. Adenovirus is used as vector. That is the reason why I choose to focus, make an attempt to explain about adenovirus as a vector, because this adenovirus is used as a vector for COVID vaccine preparation. Recently, COVID affected crores of lives all over the world, and we have got so many vaccines that are released into the market, like Covaxin, Pfizer. Likewise, many vaccines are produced, released into the market, and these, in the preparation of COVID vaccine, adenovirus is used as vector. So adenoviruses are non-enveloped double-stranded DNA viruses with a genome size of 34 to 43 kb. And in humans, adenovirus generally cause mild respiratory gastrointestinal tracts. Adenovirus generally we use for common cold. They cause infections like fever and common cold. Jalubu antam kada. Adi adenovirus valla. And these viruses are isolated from a wide variety of mammalian species, ranging from simians to chimpanzees to human beings. In the human being itself, more than 50 varieties of adenoviruses are present, are identified. In the adenovirus, there are two types of genes. In the adenovirus, there are two types of genes, early genes and late genes. Early genes are named as ABC, like EA1, EAB, E1B, E2, E3, E4, early genes, because they are very essential or necessary for supporting viral replication inside the host cell. They are called early genes. And late genes are also present. And these genes are required for host cell lysis, viral assembly, and virion release. And this adenovirus can able to carry more than 45 kb of lengthy DNA. So that is about vectors, right? So we have covered the cloning vectors, the expression vectors, vector used for bacteria, yeast, vectors for animal cells, plant cells, prokaryotic cells, eukaryotic cells. There are different vectors. As I told you earlier, the purpose of vectors to produce vaccines, every vaccine today are produced by vectors. And especially diabetes, we use insulin hormone. Once upon a time, we used to use insulin taken from pig to treat the diabetes. But now, the human insulin is produced by using vectors. Vectors are used to transfer this gene from human being to the bacteria. And now through the bacteria, we are producing insulin, which is called humulin, because the origin is from human. So vectors are used in hormone productions like insulin, growth hormone, thyroid hormone. Different hormones are there in our body. And all the hormones and all the enzymes in our body now produced in the laboratory with the help of vectors. And these vectors are also useful in brewing industry for the production of beer, for the production of alcoholic beverages. Vectors are used. And vectors are used for recombinant DNA technology, especially to produce genetically modified organisms. We have genetically modified plants are there, genetically modified animals are there. In all these experiments, vectors are used. What not every product that you see, every hybrid variety, every hybrid plant that you see in the market today, hybrid varieties of like rice, golden rice we are using right now. Golden rice is a very rich in vitamin A, where the gene that responsible for vitamin A from the carrot is taken and introduced into the rice, and now that rice is called golden rice. So how this is possible? This is all possible due to vectors. We use some vector, some bacteria, some virus, some plasmid or phage to transfer our desired genes. And genetically modified organisms and drought resistant varieties for plants, disease resistant varieties for plants, all these are produced with the help of vectors. Thus, vectors play a very important key role in the present biotechnology. And they're very useful, especially in building a genomic libraries, especially human genome project. And now scientists are doing research, collecting the genome libraries, are producing genome libraries for every organism on the earth. And vectors are used to clone the DNA, multiple copies. Likewise, these vectors have a vast role in biotechnology. So that's how we covered varieties of vectors, especially focusing on your syllabi. And one more thing, we have got bacterial artificial chromosome, BAC, and yeast artificial chromosome, BAC and YAC. That's also important, where the entire chromosome, very lengthy chromosome, the lengthy DNAs can be transferred with the help of bacterial artificial chromosome and yeast artificial chromosome. So that's about. With this, I'll conclude my webinar on the role of vectors in biotechnology. And now I invite the student community to ask any doubts regarding this.
All the participants are requested to raise their doubts. My dear participants, do you have any doubts? Ravinder sir, ready to clarify your doubts?
Recording in progress. Participants,
Sir Dattu garu.
Hello, sir.
This is Ramesh, sir.
Sir,
Please explain about the difference between cloning and expression vectors, sir. What are the differences between expression vectors and cloning vectors? What are the additional requirements for expression vectors?
yeah. In case of cloning vectors, clone means we get multiple number of copies. Okay. For example, to produce gene libraries, there is no expression. They just clone the DNA, they pack it in a vector, and they place it one particular place. There is no expression of gene. There is no production. We simply take the DNA, we pack it in a vector, we store it one place. Whereas in expression vector, you allow that gene to express of course, both function is same. The expression vector in the sense that DNA is allowed to express, transcript, translate, and to produce some product.
Thank you, sir.
Participants, any more doubts? Datta sir, any questions that is in the chat box, sir?
No, didn't see anything, sir. Chat box. Which vector has the highest cloning capacity?
Sushma, she is asking. COVID vaccine is not a vaccine, it is a immunity booster. Can I get the explanation about it, sir?
Yeah, the one point she raised is correct, because vaccine means it must give protection, right? For some strains, COVID is acting as a vaccine in the sense it giving protection, but the COVID vaccine is continually changing its genetical format. So some vaccines are not giving protection for some strains of vaccine. Actually, still it is in research. No perfect vaccine is produced for COVID. That's the reason why in the second phase, even though some people got vaccinated, they got COVID, but the intensity is less. In that sense, we can call it as a immunity booster. For some strains, this is acting as immunity booster. Yes, you said is right. It is an immunity booster. But at the same time, for some strains, it is giving complete protection. In that case, it is acting as a vaccine. You're right, Sushma. Both can be explained because it's still in research form.
Thank you, sir. One more question, but that's from Harsha.
Which vector has the highest cloning capacity? Yeah,
Which
Harsha.
organism has the highest number of vector? Why do you plasmid needs to be very small, like in KB?
Where is that question, sir?
From Harsha, sir, that is in the chat box.
Where? Can you read that question, sir, once? Can you read that question one more time? Hello, sir. Sir, your voice is not audible.
Once again, sir. Which organism has the highest number of vector? Why do you plasmid needs to be very small, like in KB?
Yeah, because the length of the DNA, unless it is small, it cannot be transferred. Because the length of the DNA is very small. No, that is the reason why the plasmid must be small. And based on the size of the DNA, we use different kind of vectors. Whereas in BAC and YAC, the vector is the size is very large, because at that bacterial artificial chromosome, we transfer a lengthy DNA. That's the reason why BAC and YAC, their sizes are very big. Whereas if the size of the DNA is small, we just use like plasmid. That's it.
Thank you, sir. Thank you, sir. Some participants are requested to upload the entire material for their preparation or the.
Sure, sir. I'll share I'll leave the PPT in the group.
Thank you. Thank you so much, sir. Thank you so much.
Group.
Any more questions from participants? Thank you so much, sir. Dattu sir.
Thank you, sir.
On behalf of our principal, sir, is also received the one message from the important message, sir. That's why just he is leaving campus.
Okay.
I would like to give the concluding remarks, sir. On behalf of S.V.K.P and Dr.K.S. Arts and Science College Management, Principal, staff, and students, and especially organizing the departments of the Department of Zoology and Biotechnology, I convey my cordial thanks to Pravin Dattu sir for elaborately explaining the vital role of the vectors that is in the biotechnology. You are also explained pinpointly, definitely that is also useful to all the participants, sir. Really, all the science students, even though the language people like ours, who are also enjoyed a lot, sir. Thank you once again. God bless you. God may give us the good strength and vision that is the future, regarding the future explanation regarding the some of the nice topics. If there is any possibility, we would like to arrange the next academic year one seminar, sir, not the webinar. If you are giving the permission to us, we would like to arrange one seminar to our faculty and students also.
Sure, sir. Sure.
You permit
Thank you
me,
very
sir,
much.
next academic year?
Sure, sir.
Thank you. Thank you. God bless you, sir.
Thank you very much.
Now, I would like to invite head of the Department of the Biotechnology, Sri T. Ramesh, sir, for propose the vote of thanks.
Good afternoon, ladies and gentlemen. On behalf of S.V.K.P and Dr.K.S. Arts and Science College, management, principal, staff, and students, I propose a vote of thanks at the successful completion of the national webinar on vectors and their role in biotechnology, jointly organized by Department of Zoology and Biotechnology. At the outset, I express my sincere thanks to Dr. Y.V. Apparao Garu, Principal of the college and Chairman of the today's webinar, for his valuable opening remarks and blessings. I am very thankful to the S.V.K.P Garu, Head of the Department Zoology and Coordinator IQAC, for his blessings and encouragement. I cordially thank Dr. K. Lakshmikantam Garu, Department of Zoology and convener of the webinar, for her keen interest in arranging the webinar, in which it is very successful present, it is very useful for the present scenario. And express my sincere thanks to the resource person of today's webinar, Dr. Pravin Dattu Garu, for sparing his valuable time to deliver his inspiring, thought-provoking lecture. I also convey my thanks to faculty members of UZ, PZ, Zoology and Biotechnology for their kind support in organizing the webinar. I am thankful to Sri M. Sairam Garu for extending his technical support throughout the webinar. And finally, I express my deep, sincere thanks to the college management, principal, staff, and students, and in particular participants from the various corners of states, extending their wholehearted support and monumental patience for the successful completion of the webinar. Thank you, one and all. Thank you for giving me this opportunity.
Thank you, sir.
Thank you so much, Ramesh Garu. Thank you so much. Finally, I convey my cordial thanks to all the participants, especially Pravin Dattu sir, for spending time with us. Thank you so much. Participants, we're already posted the feedback on the chat box. Don't post it, that is in your WhatsApp group. First, you have to receive the certificates. Then tomorrow, we would like to post the certificate feedback form to all the participants in our WhatsApp group. If you are posted, that is in the WhatsApp group, you may not get the certificates today. Only 100 certificates are generated today. That's why who are receiving that is feedback form that is in the chat box, fill it immediately and submit it. Within seconds, you are able to get certificates. Thank you one and all. Ravindra Devaya, thank you so much
Thank you.
for wonderful message. Ruth madam,
you, sir.
do you want to say?
Thank you very much. So
Thanks,
for your
Amma. Thank you
excellent
for this.
speech, thank you, Anna.
Thank you for the
Thank you very
opportunity.
much. Even though you are in busy schedule, so you are accepted for my invitation. Thank you very much, Anna.
Thank you, Ra. Thank you very much.
మన టాబ్లెట్ ఎప్పుడు కొనుక్కుంటాం?
Sorry, అదేనా హ్యాండ్ అవుట్ చేసుకోవాలి మనం అన్న. Lattu sir, please continue. 1 minute.
Okay. Okay, sir. Okay, sir.
And close చేసేస్తా.
So you can close. Hello, దత్తు గారు.
సార్ ఎవరు సార్?
నేను భూషణ్ అండి, విశాఖపట్నం.
సార్ నమస్తే భూషణ్ సార్, బాగున్నారా?
ఎక్సలెంట్ సార్, ఎక్సలెంట్.
థ్యాంక్ యూ సార్ మీరు జాయిన్ అయినందుకు సంతోషం. సార్ మన స్టూడెంట్స్ జాయిన్ అయ్యారు సార్ భూషణ్ సార్.
జాగ్రత్తగా వాడుకోవాలి, అక్కడ భోజనం తిరగడానికి చూసుకోవాలి. ఇక్కడే వాడుతుంది.
ఎవరు సార్?
Thank you very much.
Hello sir. Thank you very much.
Very useful session. We expect one more session very shortly.
That is seminar.
Prabhavaram sir. Okay sir. Thank you. మీరు సమ్మర్ లో
We don't want any webinars, but we want your valuable presence in our college.
Sure sir, sure. చాలా సంతోషం మిమ్మల్ని అందరిని చూడటం ఈ విధంగానైనా. కృపవరం సార్, శశి గారు, రమేష్ గారు. హలో సార్, రమేష్ గారు బాగున్నారా? చాలా మారిపోయారు.
మా యొక్క సపోర్ట్
సాయిరామ్ మొత్తానికి యూట్యూబ్ ని సక్సెస్ చేశారు లాస్ట్ మినిట్ లో అయినా. సరే, స్టాప్ ద రికార్డింగ్. ఎక్కడిది?
ఓకే భయ్యా,
రికార్డ్ ని స్టాప్ చేయండి.
థాంక్యూ సో మచ్ వన్స్ అగైన్. గాడ్ బ్లెస్ యు.
ఓకే సార్, థాంక్యూ. ఓకే రైట్. థాంక్యూ.
దివాకర్ భాయ్, దివాకర్
సార్, ఇది రికార్డింగ్ స్టాప్ చేయటం ఎక్కడ?
ఓకే సార్. ద బాటమ్ హోస్ట్ ని రిమూవ్ చేయండి. ప్లీజ్ రిమూవ్ యువర్ హోస్ట్ సార్.
హోస్టా
సార్? అక్కడ మీ నేమ్ తిన్నగా మేక్ మీ హోస్ట్ ఉంటుంది సార్. అది
ఎక్కడండి? నా పేరు దగ్గర, నా వీడియో దగ్గర
పనిచేయండి. మా S.V.K.P ఉంది కదా సార్, అక్కడ మా S.V.K.P దగ్గర రైట్ క్లిక్ ఇచ్చి మేక్ ఏ హోస్ట్ అని క్లిక్ చేయండి. అప్పుడు సార్ పార్టిసిపెంట్స్
మీ వీడియో ఉందా? మీ మీడియా మీద
S.V.K.P దగ్గర సార్.
S.V.K.P
ఫైన్ సార్. ఇప్పుడు పార్టిసిపెంట్స్
రికార్డ్ స్టాప్ రికార్డింగ్ చేశాను.
రైట్, గాట్ ఇట్.
రైట్ సార్. శశి గారు, కృపావరం గారు, అదే స్మార్ట్ సేమ్ తగలా.
అక్కడ రైట్ ఇప్పుడు సార్ S.V.K.P ఉంది కదా సార్, అక్కడ మోర్ దగ్గర రైట్ క్లిక్ అవ్వండి సార్. అక్కడ మోర్ దగ్గర క్లిక్ చేసి మేక్ మీ హోస్ట్ అని కొట్టండి సార్. అక్కడ
ఎక్కడ సార్ అది? మీ దగ్గర
మీ పేరు పైన సార్ పార్టిసిపెంట్స్ క్లిక్ చేయండి సార్. అక్కడ
క్లిక్ ఆన్ ది పార్టిసిపెంట్స్.
పార్టిసిపెంట్స్.
ఓకే, దెన్ S.V.K.P ఇక్కడ మోర్ అనే ఆప్షన్ కదా సార్, దెన్ యూ కెన్ రైట్ క్లిక్ అండ్ మేక్ మీ హోస్ట్.
మ్యూట్ అండ్
మేక్
మోర్.
మీ హోస్ట్.
Right, you can make it sir. I'll get access.
ఓకే సార్, కట్ చేశాను, రికార్డింగ్ స్టాప్ చేశాను.
ఓకే ఫైన్ సార్. రైట్,
ఇప్పుడు రికార్డింగ్ షేర్ చేయాలా మీకు?
నాట్ రిక్వైర్డ్ సార్, మాది ఆటోమేటిక్ మా దాంట్లో సేవ్ అవుతుంది.
Okay, sir. Thank you.
Okay, thanks.
Thank you.
Thank you, Bhaiya.
Thank you very much.
Thank you so much. Sorry.